Okay. So this slide is all about the isoforms of LOX, isoform of lipoxygenase genes. So what I have told that there can be different isoforms of this lipoxygenase gene. Now these genes of this lipoxygenase enzyme are basically members of multigene families. So in different plants there can be different isoforms of this gene. Like in case of Arabidopsis thaliana, you can get 6 isoforms of lipoxygenase. In case of potato, you can get 14 isoforms of lipoxygenase. So, as I have told that these polyunsaturated fatty acids act as the precursor of these lipoxygenase enzymes. So, this one example I have given where different isoforms of LOX actually act on these polyunsaturated fatty acids. In case of angiosperms, the main polyunsaturated fatty acids are linoleic acid and alpha linolenic acid. So this 18 carbon fatty acid with two unsaturation that this is the linoleic acid. On this fatty acid, lipoxygenase will work. Now these 9 logs and 13 logs, that 9 and 13 actually representing at which position of this fatty acid chain, at which carbon position of this fatty acid chain the hydroperoxygenation will occur. Now 9 lipoxygenase or 9 logs will work on this fatty acid chain at the ninth position of this fatty acid chain, and 13 logs will hydroperoxygenate at the 13th position of this fatty acid chain. In this way, with help of 9 lipoxygenase, you will get 9 hydroperoxy fatty acids. With help of 13 lipoxygenase gene, you will get 13 hydroperoxy fatty acids. Now, these two hydroperoxy fatty acids or the other hydroperoxy fatty acids formed by different isoforms of lipoxygenase will now act as the precursor for the other enzymes. Now, the other enzymes will work on these hydroperoxygenated fatty acid, and they will form different other metabolites. In this way, on the 18 3 fatty acid means alpha linolenic acid can also act as the precursor for other enzymes. Apart from this lipoxygenase, another enzyme is there, but this enzyme is not that much of dominant like that of lipoxygenase. This is alpha dioxygenase. Alpha dioxygenase enzyme is basically hydroperoxygenating at the second position or the alpha carbon position of the fatty acid chain. So the second position of the fatty acid chain is hydroperoxygenated with help of this alpha dioxygenase, and this enzyme can further be modified to other oxylipins. So as I am working with mosses, so I have a special interest on mosses and their fatty acid profiles. So in case of mosses, unlike like that of this 18 carbon fatty acid in angiosperms, these mosses are mainly contains this longer chain fatty acids, means 20 carbon fatty acid, polyunsaturated fatty acid. In this case, arachidonic acid is the main fatty acid. Arachidonic acid is the main fatty acid on which lipoxygenase will work. In mosses, apart from 9 and 13 lipoxygenase, there is 12 lipoxygenase. Means 12 lipoxygenase will hydroperoxygenate at the 12th position of the fatty acid. So 12 lipoxygenase will hydroperoxygenate the 12th position of the fatty acid. So on this 12 hydroperoxygenated fatty acid, now hydroperoxide lyase will work. This HPL enzyme will work. Now the function of this HPL is that this HPL will cleave the hydrocarbon chain at the position where hydroperoxygenation has occurred. So HPL will cleave the hydrocarbon chain at the position where hydroperoxygenation has occurred. That is why you will get two different carbon chain length compounds. So you will get 9 carbon one compound and 11 carbon another compound. So this type of metabolites will altogether form, and all these compounds will constitute these free oxylipins. So if we consider algae, in algae also different types of metabolites you will get. there are different reports on macroalgal members. So there are main fatty acids in case of algae also, 18 carbon fatty acids and 20 carbon fatty acids. All these polyunsaturated fatty acids will act as the precursor for these metabolites. So these all these examples are given, means these examples, these all compounds which are formed from linoleic acid. So alpha linolenic acid means 18 carbon another fatty acid, stearidonic acid that can also act as the precursor. In case of 20 carbon fatty acid, arachidonic acid and icosapentaenoic acids both will act as the precursor. Even 22 carbon fatty acid, which is docosahexaenoic acid, that can act also as the precursor for these compounds. So all these fatty acids will act as the precursor, and you may expect a variety of metabolites can be formed with help of lipoxygenase and the subsequent enzymes, and all together they will constitute oxylipins in algae. Now, free oxylipins are basically present in the soluble compartments, main cytosol or stroma of plastids or peroxisomal matrix. And these modified esterified lipids means the oxylipins which are esterified with glycolipids or phospholipid. One example I have told, that is arabidopside. Arabidopside is one very important compound that is formed, where, yeah, I have already told that these modified fatty acids are attached at their SN1 and SN2 position. So this compound is now known, and it is some researchers are working on. So but their significance is not that much of clear till now. So, if we talk about the significance of these compounds, so I have already told about the structural details. Now, coming into the significance part. So, what I have told already, that these oxylipins are mainly generated in higher amount due to biotic and abiotic stress responses. So, one example I have given, where two volatile chromatograms are given here. You can see two volatile chromatograms. One is the control one, another one is the same plant which is infested by one herbivore. So you can see that in this infested plant, if the in the chromatogram there are some volatiles which are present in very high amount. These are called infested plant oxylipin generation is high amount. So this is visible.
Hello audience, sorry for the interruption. We'll start the session after 1 hour. Okay.